My name is Oscar Monje, a member of the space crop production team at the Kennedy Space Center. My co-authors are Lachelle Spencer, Matthew Nugent, Matthew Romine, and Ralph Ritchie. I will discuss space farming, a sustainable crop production technology. In space, explorers need in situ crop production. Space farming enables colonization of space. Technologies must be sustainable, meaning they minimize the logistics of resupply. They provide resources for plant growth, light, CO2, oxygen, nutrients, water, and seeds. And plant chambers like LADA, BPS, and veggie have been used to grow space crops, but new plant chambers with improved watering systems are needed. Plants also provide the crew physiological well-being by providing green earth smells and tastes to the crew. They also supplement current food systems with palatable, nutritious, and safe sources of fresh crops during long-duration missions. The ISS food system is adequate for low Earth orbit missions. However, processed and prepackaged food loses nutrients, for example, potassium, magnesium, vitamins, during extended storage, and may not provide the expected nutritional content or have the same taste when consumed. Thus, NASA has identified robust pick-and-eat crops, for example, lettuce, radish, and peppers, for supplementing crew diets with fresh leafy greens during near-term low Earth orbit, cislunar, lunar, and Martian missions. This approach mitigates the risks of inadequate food and nutrition during long-duration NASA exploration missions. New challenges of space crop production in reduced gravity are demonstrating soilless gravity-independent water delivery systems, safely growing salad crops, and identifying new crops adapted for growth in spacecraft. These solutions will enable Artemis long-duration exploration missions that include Martian missions taking over 3 years. Space production systems will also be needed to support a growing commercial space economy that will essentially increase the crew size in cislunar space. In this cislunar 1000 plan, it's envisioned that in 5 years, the population of humans in space will grow to 20 people. That is as space manufacturing, commercial habitats, and other prospecting missions become available. And then it's even expected to grow up to 300 in crew members in the next 15 years as you increase tourism and low Earth orbit and other propellant and lunar propellant mining operations on the moon. As an example of this development, we already seen the development of the Axiom space station, a habitat and a commercial space and research and manufacturing platform to be deployed soon. Thus, new life support concepts like space crop production must be demonstrated at a scale to sustain large crew sizes in cislunar space. Szabo cataloged the last 50 years of spaceflight hardware. This graph captures how plant systems have evolved. Missions on the Salyut, Mir, Space Shuttle, and ISS were conducted in plant chambers with small planting areas until about 2010. Space biology experiments from the 1970s to the 2000 were conducted in small plant growth chambers that provided low light levels, and between 2000 and 2015, environmental control improved in these chambers and light levels increased. So these are recent advances in technology. These experiments identified that indirect effects of microgravity reduce plant growth in low Earth orbit. The absence of buoyancy-driven convection causes poor mass and heat transfer to leaves and plant organs, and capillary-driven moisture redistribution in the root zone can lead to poor root zone aeration. In the last 10 years, two large, around 0.2 square meter in area, space crop production systems, the veggie and the advanced plant habitat, were developed by NASA. Veggie provides 300 micromoles per meter squared per second of RGB LED light, has minimal environmental control, and uses a passive watering system. In contrast, APH and the future Ohalo 3 provide full control of the cultural environment and can operate at high light, less than 1,000 micromoles per meter squared per second. Recently, leafy vegetables grown in veggie on ISS were found to be a safe, edible source of fresh crops to supplement the astronaut food system. The Advanced Plant Habitat Facility on ISS is a platform for conducting crop growth experiments lasting a whole life cycle, around up to 150 days on ISS. In 2017, the first plant test was a tech demo for hardware and science validation of the Advanced Plant Habitat Facility. Both Arabidopsis plants and wheat plants were grown for 30 days to put a load on the system for moisture control and for CO2 control. The Arabidopsis growth system was adopted in the PH01 peer-reviewed research funded by NASA with the PI Norman Lewis, and entitled An Integrated Omics Guided Approach to Lignification and Gravitational Responses, the Final Frontier. The experiment consisted of growing Arabidopsis in space and collecting samples for omics analysis. Currently, the PH02 mission is ongoing. The PI is Carl Hassenstein, and this research is entitled Assessment of Nutritional Value and Growth Parameters of Space-Grown Plants, and they're growing radishes. In 2021, PH04, another technology demonstration, will be launched. The PI is Matthew Romine, and the experiment is entitled Growth of New Mexico Hatch Green Chili as a Technical Display of Advanced Plant Habitat's Capabilities. The images here show the ground studies conducted prior to deploying the 120-day-long mission on ISS. We have to develop the planting protocols, seeding, tending the plants. Here's a 28-day event where plants are thinned, and then eventually plants grow to 74 days and 116 days. And these are some of the examples of the peppers that are produced. Currently, we're transitioning from space biology to crop production in these technologies. The solid substrate-based water delivery system used in the APH, shown here on the right, evolved in the SVET, Astroculture, ADVAS, LADA, and BPS chambers. However, since plants are grown for science, the APH root module is launched pre-planted, shown here, containing 4 kilograms of substrate, roughly 1 kilogram of substrate per each of the 4 quadrants, and is discarded after the plants are harvested. These practices are unsustainable for space crop production systems. For example, scaling from the 0.2 meter squared APH rooting system to a salad machine producing 13 outrageous lettuce crops per year, produces 5.3 kilograms of lettuce, but requires about 53 kilograms of fertilized medium. Thus, the APH system has a low productivity ratio of 0.1 kilograms of edible mass per kilogram of resupply mass. Therefore, new soilless water and nutrient delivery systems are needed to avoid constant resupply of bulky single-use porous media. This will improve sustainability of crop production systems. Hydroponics, the nutrient film technique, is a leading technology for optimal food production on Earth. The APH substrate-based system, aeroponics, and the omni-gravity hydroponic system are alternates developed to address challenges for containment of the water, providing sufficient aeration to the roots, and addressing liquid gas separation observed in microgravity. Recently, the productivity of two soilless candidate water nutrient delivery systems, the porous tube nutrient delivery system PTNDS and the on-demand system, were compared to the productivity of NFT and APH. The PTNDS utilizes ceramic porous membranes held under suction for watering plants germinated directly on the tubes. Here's the tube shown here, and the plants are grown on them. And the on-demand system uses a feedback from a moisture sensor to maintain a constant root zone moisture. The NFT system produced on average 78 gram plants, and the productivities of the APH, on-demand, and PTNDS system were 51%, 39%, and 34% of the NFT respectively. These results suggest that water delivery technologies that close these gaps in productivity are also needed. Autonomous plant health and food safety monitoring systems are needed to ensure that fresh crops produced in future space crop production systems are safe for crew consumption. Future autonomous crop production systems may be outfitted with imaging systems to capture plant, hyperspectral, thermal, and fluorescence data in near real time over their entire growth cycle. These systems may respond by raising alarms when stressed due to nutrient deficiencies, drought, flooding, or microbial or fungal infections are detected. Thus, monitoring plant health for food safety could be achieved with minimal crew intervention. New plant species and cultivars with improved growth habits and contents of antioxidants, vitamins, and minerals are also needed when grown in spaceflight environmental conditions. These are typically super elevated CO2 concentration with ultra-spectral recipes. Veggie cultural selection efforts at KSC have found that the environment affects germination, growth habits, biomass production, nutritional value, and even flavor of plants. Thus, plants grown on ISS conditions or selected under these conditions of 40% relative humidity, a veggie spectral quality, and elevated CO2 have higher crop readiness levels for use in veggie. This will ensure their success when deployed in microgravity. These cultivar selection has also studied the yield responses of leafy greens, lettuce, pak choi, shunjiku, Swiss chard, mustard, and kale to supplement far red light. Responses included increased plant canopy cover and increased shoot height, which may be a consideration for volume-constrained space crop production systems. In summary, new microgravity-independent water and nutrient delivery systems, autonomous plant health monitoring technologies, and improved crops will be needed to enable crop growth demonstrations on Artemis lunar landers and habitats. Also needed to supply pick and eat crops to crew food systems deployed on Mars transit missions, and for studying the effects of combined partial gravity and deep space radiation on crops grown on the moon. This work was funded by NASA's Biological and Physical Sciences Division. Thank you.